Hi, everybody, and welcome to this week's lecture. What we're going to do for this week is we're going to go ahead and introduce biologic molecules, also known as macromolecules, and we're going to go ahead and kind of break this down into four parts. We're going to look at the introduction today. We're going to go ahead and look at our proteins, carbohydrates, nucleic acids, and lipids in the following ones. So let's go ahead and get started and see exactly what we're going to try to accomplish for this lecture. So by the end of this week, you should be able to identify all of the different types of biological molecules. In this case, we have carbohydrates, proteins, lipids, and our nucleic acids. What you're also going to be able to do is you're going to be able to understand two major reactions. The first one is going to be dehydration synthesis, which in this case just means the absence or the subtraction of water. And synthesis is just the production or making or creating something. And then our anabolic chemical reactions. And we're also going to look at the opposite end, which is hydrolysis, which is going to be the addition of water. And then we're going to look at our catabolic chemical reactions. So before we go ahead and do all that, what we're going to do is we're going to identify the four major classes of macromolecules. Now, before we do that, let's go ahead and break down this word, macro and molecule. So macro, in this case, just means big. So I'm going to represent that with a plus. And then our molecules, just like in our previous lectures, are going to be a bunch of atoms or elements coming together in order to produce something much larger. So in this case, it's just going to be large molecules. Now, you'll notice down here we have a visual, and this visual is kind of showing us exactly what elements are going to comprise in the human body. You can see we have many different types over here. We have oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus. We have potassium, sulfur, sodium, chloride, and magnesium. And then we have other trace ones. And the reason why these are so important is because all of these together in certain combinations are going to produce our macromolecules. And our macromolecules are going to be carbohydrates, also known as carbs. We have our lipids, also known as fats, our proteins, and then our nucleic acids. Now, when it comes to all of these are all going to be important and essential for life. These also are going to be known as organic molecules. And the reason why they're going to be known as organic molecules is because each one of these are going to contain carbon. So again, all these together are going to contain this element here. And carbon is often known as the element of life. Now, these are also going to contain other elements such as hydrogen, oxygen, nitrogen, and other minor elements. But again, every single one of these is going to contain that carbon. Now, water is going to be an example of an inorganic compound. Okay, so anytime an organic molecule is mentioned, it means that it contains carbon. As you know, water is going to be H2O, and you can see that it does not have any carbon in it, meaning that this is going to be considered an inorganic compound. Okay, so to kind of summarize this slide, we have our four major macromolecules. Remember, macro meaning big, molecule meaning those separate pieces, macro, big molecules, adding all those together in order to produce our four macromolecules right here, which are our carbohydrates, our lipids, proteins, and nucleic acids. All of these are going to be considered organic molecules because they contain that carbon. Water is an example of an inorganic compound because it does not contain any carbon in it. So now that we've went ahead and identified that, what we're going to do now is see exactly why they're so important. What are the main functions of these macromolecules? Why do we need to know them? And why are they important to you? So when it comes to that, what we need to do is identify this top part here. Our macromolecule, remember macro meaning big molecules. So our four, again, are carbohydrates, also known as carbs, proteins, lipids, which are known as fats, and then our nucleic acids. Our monomer is going to be another very important vocabulary word. So when we're dealing with macromolecules, they're going to be made up of many different parts. We have our monomers, our polymers, things like that. Now, when it comes to our monomer, all a monomer means is, just like the name suggests, mono, meaning one. These are going to be our smallest denominators, meaning that these are going to be our most small and basic forms of our macromolecules. Okay, so mono, one, smallest denominator when it comes to our macromolecules. Function, just like the name suggests, is what's the point of it? What is it going to do in our body? And then some examples. So let's go ahead and take a look at carbohydrates to start off with. In carbohydrates, our monomer, or in this case, our smallest molecule for this, is going to be known as a monosaccharide. Mono, meaning one, saccharide, meaning sweet. So in this case, monosaccharide is going to be our carbohydrates. It's going to be our smallest denominator or our smallest form. Some examples of monomers are going to be glucose and fructose. So in this case, glucose would only be made out of one piece and fructose being one as well. But we'll go ahead and see more examples of this as we continue. In our function, we have two different types. Our first one is going to be energy. When it comes to carbohydrates, if you ever have done any running or any physical activity, you might typically want to carb up, meaning you're going to increase the amount of carbohydrates in your body because it's going to be a great source of energy. Now, it can provide structural support as well. Typically, this is going to be found in plants. Okay, so typically that's going to be in the form of cellulose. And then again, examples of carbohydrates are going to be starches, so anything that contains kind of bread or starch, and then sugars. Proteins are going to be another example of a macromolecule, and the monomer, so remember the smallest form of proteins are going to be amino acids. Okay, functions of proteins are going to be enzymes, which remember enzymes are going to be proteins that speed up reactions. So great examples are going to be lactase. So if you know someone who is lactose intolerant, they do not produce the enzyme lactase. So that means that when they drink milk or have any sort of dairy product, they typically become bloated and don't feel very well. And the reason why is because they do not have the enzyme lactase to break down lactose, so they end up getting very bloated. It's also for structural and movement. So in this case, our muscles to help us move around, do things like that, regulation, and cell communication. And you can typically find these in muscles as well as hair, specifically keratin. Our next one is going to be our lipids, also known as fats. And our monomer, so the smallest version of this, is going to be fatty acids. Fatty acids are going to be great for cell membranes. It's also going to be good for storing energy as well as insulation, keeping us warm. And it's going to help produce our steroid hormones. So in this case, examples are going to be adipose tissue cells. And in this case, this is just a scientific way of saying fat cells. And again, remember, it's great for storing energy and it's great for insulation. Our final macromolecule is going to be our nucleic acids. Our smallest are going to be our nucleotides, which are going to be comprised of our phosphate, our deoxyribose sugar, as well as our nitrogen base. And this is going to be our genetic material, or in this case, DNA. And as you know, that's going to be incredibly important because that is going to be the blueprints to our body. So now that we went ahead and took a look at our main functions for our macromolecules, the next thing we need to identify is exactly how do living organisms break down macromolecules? Because remember, macro means big, big molecules. So we have a way of breaking this down, and what we're going to utilize is going to be water, H2O, which is going to be incredibly important because we're going to go through something known as hydrolysis. What that essentially is we're taking a big piece and we're trying to break it down into two separate pieces. So in this case, we have maltose. Maltose is going to be known as a disaccharide. The reason why it's a disaccharide is because you can see we have 1 and 2 pieces here. So di, just like a pair of dice, are going to be 2. And we're breaking these down using water in order to create our 2 monosaccharides. 1, 2, mono. And you can see we have 1 and 2. So the way that this happens is you can see that our oxygen right here is what's holding together these 2 pieces. Now, once we add water, which remember is our inorganic compound, that is going to go ahead and break that bond. So we're going to have our OHs on each side. And because of that, it's going to break it apart, turning our disaccharide into two monosaccharides. So we went from one piece of maltose to two pieces of glucose. Okay, so again, this chemical reaction is going to be known as hydrolysis. This is where energy is produced, and water is going to be used in order to produce this. One way to remember this is hydrolysis sounds like hydro, which means water. Okay, and again, what's happening is we're catabolizing these disaccharides of maltose into two monosaccharides of glucose. So let's go ahead and take a look at the opposite end of this. So how are we going to synthesize macromolecules? Because sometimes when we consume and eat things, we don't always necessarily have the components that we need. So our body will go ahead and create or make those. Now, one thing you need to know is the word synthesize. All synthesize means is to make. So we're making something. So the way we're going to do this is we're going to go through something known as dehydration synthesis. So just like the name suggests, when you are very, very dehydrated, it is going to be the absence of water. And then synthesis is going to be to make. So we're going to be making something by taking away water. So in this case, you can see we're using the exact same example. You can see we have 1, 2 of glucose. Remember, glucose is going to be our monomer, and we're trying to produce one piece of maltose, which again, we have 1, 2 monomers coming together to make a disaccharide. Remember, disaccharide is 2. So what's happening here is we're going ahead and we're taking this piece out. We have 1, 2 H's and 1 O, so that's going to give us H2O. So that is going to be removed. And now we have that oxygen binding back together, bringing these two together and removing that water from that equation. So this is how we're getting our two glucoses to come back together to produce one maltose. So again, in this case, it is the chemical reaction called dehydration synthesis, and energy is going to be needed, right? Because we need that energy in order to kick this water out, and water is being removed. So again, in this case, we're taking those two glucoses, or our monosaccharides, and we're adding both of those together to produce maltose. All right. And the last final idea before we go ahead and test our knowledge is hydrolysis and dehydration synthesis are reversible chemical reactions. All we're doing here is hydrolysis. Remember, we're just adding water. And then in a dehydration synthesis, remember, dehydration is going to be the absence of water. When you're very dehydrated, you're missing water. Synthesis, to make, so we're losing water in order to make something. All right, so that's going to conclude our lecture. What I would like you to do for our test our knowledge is I want you to go ahead and look back through our lecture and see if you can fill in these blanks. I'm going to go ahead and give everybody a couple of seconds to pause this and give this a try. All right, welcome back. So here are the answers to those questions. What I'm going to do is I'm just going to quickly touch on a couple of things just to make sure that everybody's on the same page. It's important to understand some of these prefixes. So our mono, remember, is 1, just like a monocle is just one piece of glass over the eye. Die is going to be 2, just like a pair of dice. You'll have 2 of them. And then poly is going to be many. So it's going to be at least more than 2. Okay, a great example is polydactyly. So polydactyly, typically we have 1, 2, 3, 4, 5 fingers, but polydactyly, people typically have more than 5 fingers. So again, poly is going to be many or more, in this case, than 2. So our next part's amino acid is going to be our mono for our protein right here. And then when we take a ton of these amino acids, and we're going through dehydration synthesis, so minus H2O, synthesis producing, in this case, we're trying to produce polypeptides, which in this case is going to be our polymer. Many more than two are going to produce our proteins. Then finally, nucleic acids, nucleotides undergo dehydration synthesis to perform nucleic acids such as DNA. So remember, a nucleotide is going to be our monomer. Dehydration synthesis, remember, minus water. Synthesis is going to be to make. So we're taking a bunch of our monomers and putting them together to produce things like DNA. All right, and just like that, that is going to conclude our lecture for part 1. If you have any questions, comments, concerns, remember, please feel free to reach out. Best way to reach me is going to be through Canvas or through email. So again, please make sure you're asking any questions if you have any. With that being said, again, that concludes this lecture. So I look forward to seeing everybody in part 2.